So thinking about the soil environment, things like pH, salinity levels, soil temperature, water content, and aeration, what food or nutrient sources are available to pathogens and other organisms, and then what other organisms are in the soil that might interact with the pathogen. And so, again, these are ones that make up a big part of the things we consider with soil health. There are also things that are very dynamic and can be sometimes hard to control in the field. So it's one reason, all of these factors influencing disease incidence is a big reason why sometimes in studies that are done in the lab or the greenhouse in very controlled settings, we might see impacts of disease suppression that we don't see in the field where these factors might be more dynamic or harder to control. So I want to talk a little bit about kind of these theories of suppression and different types of suppression as well. So there's a continuum of general to specific suppression. And general suppression is really kind of the idea that the overall microbiome and its activity is inhibiting pathogen growth and activity. So it could be potentially broad spectrum relating to a variety of soil-borne diseases. It tends to be enhanced by practices that increase the microbiome community size, activity, and or diversity. And it's been shown that increasing soil organic matter may increase general disease suppression. And so this is where, again, where soil health management comes in, is when we think about soil health management, we are often thinking about practices that will also, increase microbial activity, size, and diversity. And so in theory, this would also increase general soil-borne disease suppression. And soil-borne disease suppression tends to be reduced by soil sterilization because you're kind of knocking out a lot of the microbial community. And so how this works, the idea is that a lot of pathogens are in the soil in dormant forms, and so they're responding to root exudates, sugars and amino acids and other nutrients that are in that root rhizosphere area. And so when they detect those, then they might germinate and start to become active. But if the general microbial community is basically consuming all of those sugars and amino acids coming out of the roots, then the pathogens may not be able to detect them as readily. So it can kind of help suppress germination of these pathogens. So that's kind of the concept behind it. Specific suppression, on the other hand, is the direct attack of a pathogen by another organism. And a big aspect of this is that it can be transferable between soils with inoculum. And so I'm going to talk a little bit about some examples of this, but it's really about the population of a specific organism rather than an overall community. So maybe less affected by nutrient additions or organic matter additions, and more affected by the presence of a particular organism. And one thing to note is that this can tend to come up when one plant is continuously, one host is continuously in a soil. So in monocultures, continuous monocultures. And it tends to be kind of an up and down cycle. So there might be disease that comes up because of continuous monoculture that then makes the antagonistic organism increase its population. It decreases the pathogen, and there's disease suppression, and then the cycle goes up and down again. Whereas with general suppression, it tends to be maybe more stable because there's just kind of this general continued activity of the microbiome. But I want to emphasize that this is a continuum, and so it's important to think about what mechanisms along this continuum are at play with disease suppression in raspberry systems. Again, these are some mechanisms. I've talked about some of these, but just getting into it more specifically, on the specific suppression side, you can have bacterial attachment to pathogens. You can have bacteria or other organisms producing toxins or antibiotics that directly affect the pathogen. They can produce volatile organic compounds, or they can directly destroy the cells, maybe through cell lysis. And then on the general side of things, again, there's this competition for resources between quote unquote beneficial or just the general microbiome organisms and and the pathogen, competition for root infection sites, limitation of resources for spore germination. And then there can also be induction of plant defense, which is also an important mechanism of suppression. So I'm just going to briefly talk about some examples. There's been, on the general side, there's kind of a classic example of Phytophthora cinnamomi root rot in Australian avocado orchards that was shown to suppress Phytophthora root rot. And so this is in a rainforest situation, so very wet climate that tends to favor this host. But in areas where the rainforest had been cleared and avocado orchards had been planted, there was a lot of soil deterioration, soil acidification, loss of soil organic matter, and that brought up extensive root rot. But there was an orchard that maintained soil organic matter levels similar to the native rainforest, so very high at 12%, which is, we often don't see soils that high around here. But they brought in a lot of additions of poultry manure, crop residues, calcium, maintaining that pH at a more neutral level, and they did not have any root rot issues. And this, the thought behind this was that there was just, by maintaining this high level of soil health and soil organic matter, they were feeding the microbes that could then suppress the Phytophthora root rot. On the other side of things, on the specific side, a classic example is take-all disease, which happens in wheat. And there's been shown to be what's called take-all decline occurring with continuous monoculture of wheat or barley, because antibiotic-producing Pseudomonas grow and in response to increased levels of the pathogen. And it's something that has been shown to be transferable between fields, meaning you could take a little bit of soil from a suppressive field, move it to another field, and then see that there's suppression forming in that other field. So again, these are two examples along this kind of on either end of this spectrum. And as we move into the next few talks, some kind of leading questions I want to pose that we need to think about in raspberry systems are, which mechanisms are more at play? Is there more general or specific suppression going on for major pathogens and pests in red raspberry? What cultural practices enhance microbial activity and diversity and general suppression in red raspberry systems? And then also thinking about what cultural practices might exacerbate pathogens. There's a lot of focus on, maybe adding composts or adding green manures that might, could feed the microbiome and could lead to suppression, and also it can lead to, feeding pathogens and pests. So there can be issues with exacerbation that we need to consider. How are soil microbial communities functioning together to suppress pathogens? There's a lot of new tools we have to really understand the microbiome, but there's a lot we still don't understand about how different organisms are interacting with one another. So I think this is an area of research going forward that we really need to understand more and have soil scientists and pathologists and microbiologists all work together. And then also, of course, there's physical and chemical aspects that are important as well, things like soil aeration, pH that might need to be addressed that can be contributing to pathogen issues, not just the biology. So I'm sure that the next few talks will help answer some of these questions, and then we'll have a panel to then talk about what we still don't know and what questions we need to really think about going forward. So with that, I'm happy to take any questions or comments, or we can move on to the next speaker as well. Molly's giving me this signal.
I think we'll take questions. For any questions that you have for Deirdre, you can bring into the panel and she can answer them there just so that we can stay on time.
Thanks.
Thank you, Deirdre. Next up.